Professor Carafit here to walk you through the online photosynthesis lab. The basic research question is, what is the effect of wavelength on the rate of photosynthesis? So let's look at the big picture here. Let's start off with an organism, a plant, a tree. This looks like a cottonwood tree probably. It's an organism. It has organs, in this case a leaf. So here's a leaf, and if we zoom in on that leaf, we can see that that organ has tissues. And we can see that those tissues are composed of cells. So let's look at this organ, this cross-section of this leaf. So there's an upper and lower epidermis. The epidermis contains openings called stomata. The plural is stomata. Singular is stoma. These are openings that can open and shut and let gases in and out of the leaf. In the middle of the leaf is a tissue called the mesophyll. The mesophyll contains cells, and and veins. The veins of the leaf are found in this area as well. Those transport water and sugars from the roots to the leaves and vice versa. If we zoom in on the mesophyll, we can see one mesophyll cell down here. That's a single mesophyll cell loaded with these green blobs. These are organelles, in this case chloroplasts. The chloroplast is composed of a double membrane and a thylakoid membrane, and there's a liquid stroma inside. And the chlorophyll, the molecule that captures sunlight energy, is embedded in this thylakoid membrane. So what we would do is we would take a CO2 probe, which measures concentration of CO2 in parts per million, and we would shine lights of various colors on a plant and measure its photosynthetic rate. And keep in mind the formula here. 6 carbon dioxides plus 6 waters and light yield sugar and oxygen gas. So CO2 is an input, not an output of this reaction. So you'll have to think about what the data would look like if photosynthesis was happening. What would that graph look like? How would it be a positive number or a negative number if CO2 is an input? Okay, so what you should probably do is review your textbook, look at what colors of light are most absorbed by photosynthesis, and use that to help write a hypothesis. If and then. If a certain color of light is most absorbed by chlorophyll, more absorbed than other colors, and plants are exposed to, etc., etc. I'll let you write your own hypothesis. So what we would do is we would use spinach leaves if we were meeting in person. Spinach leaves from the grocery store are still alive and they still photosynthesize, and we would put them in a chamber, shine light on them, and stick a probe inside. I guess we don't have to worry about the tips on how to use the probe since we're not actually using it, but what we would do is we would incubate the plants for 5 minutes at that wavelength, and then we would run for 5 minutes, and we would get the slope of the line to see what the rate of change in CO2 concentrations. And then we would have to fan the probes out in between each run in order to return the probes to room levels of CO2. So here's an example of a setup. A light bulb is on a ring stand there. There's a plant inside of that container, and the bulb is touching that container basically. I have another video of the setup, so I'm going to skip past this fairly quickly. So there's a whole separate video you need to watch on the setup. Okay, so what you'll want to do is make a prediction, a hypothesis. So a hypothesis has your explanation and your prediction built into it. How do you think different colors of light will affect photosynthesis? The main colors you would be testing are going to be red, green, and blue, and white, and then darkness. What do you think will happen in darkness? So try to work out a hypothesis that predicts how those values would relate to one another. Okay, good luck, and I'll see you later.